we have. So in our previous classes, we have seen how ATP plays a central role in metabolism. It is regarded as the energy currency of the cell. And we also seen that a large number of endergonic reactions which are happening in a cell every moment consume a lot of ATPs, and the cell has to continuously resynthesize these ATPs. And there are two methods for this. One is the substrate level phosphorylation, which we have studied in detail in our previous class. So what is substrate level phosphorylation? There are high energy molecules which are much more high energetic than ATP itself, like phosphoenol pyruvate, diphosphoglycerate. Such molecules transfer a phosphate group directly to ADP, releasing ATP. Now we will move on to one more or the other method of phosphorylation, that is called oxidative phosphorylation. And mind you that the oxidative phosphorylation is the one which provides majority of the ATP to the cells. It's the oxidative phosphorylation, not the substrate level phosphorylation. The number of ATPs obtained in oxidative phosphorylation is far greater than those obtained in substrate level or through substrate level phosphorylation. So what is oxidative phosphorylation? Let us start studying this oxidative phosphorylation. So stop your videos, all of you. Well, oxidative phosphorylation, as the name itself indicates, should include a reaction which is oxidative in nature. So in oxidative phosphorylation, there must be something which is undergoing oxidation. The synthesis of ATP from ADP is not oxidation. It is phosphorylation. It is simply the transfer of phosphate group from some other molecule, or maybe in the form of inorganic phosphate to ADP. Inorganic phosphate has to join ADP to make ATP. That is not an oxidation reaction. It is basically a condensation reaction. Whereas oxidative phosphorylation, word oxidative is indicating something drastically different is happening here. Okay. So what is that different thing which is happening here? Please stop your videos here, all of you. Not understanding why you open your videos like this. Yes. So now there are two coenzymes by name nicotinamide adenine dinucleotide, abbreviated as NAD, and flavin adenine dinucleotide, abbreviated as FAD. What is this NAD? As I told you, they are coenzymes. And what are these coenzymes, by the way? They are the organic molecules. They're non-proteinaceous organic molecules which are required for the functioning of enzymes, coenzymes. Nicotinamide dinucleotide is simpler in structure than FAD. You know a nucleotide, right? So what is nucleotide? Basically is a ribose sugar to which joined phosphate group on the 5th carbon atom or a nitrogenous base on the 1st carbon atom. So nicotinamide is almost like that. NAD is almost like that, except it's a dinucleotide. So there are two such residues which are connected. Not ATP was a mononucleotide, whereas NAD is a dinucleotide. What is DNA and RNA? Polynucleotide. You have large numbers of nucleotides connected like this through a phosphodiester bond. So this is what the phosphodiester bond is. This is only a dinucleotide. Ribose, phosphate groups, and one nucleotide out of this two is actually an adenine. You see the adenine's nitrogenous base here. So this part represents AMP basically. The other part is different a bit, in the sense instead of the nitrogenous base adenosine, you have nicotinamide attached. You see this? This is nicotinamide. So this is NAD. Similarly, you have another coenzyme FAD, flavin adenine dinucleotide. The same thing, it's a dinucleotide. One side it is the attachment of the adenosine. The other side, it is the attachment of a moiety called flavin. This is the flavin moiety, which is attached to a sugar. Now, of course, the structure of flavin adenine dinucleotide is not required according to your syllabus, but maybe NAD structure is required. So you please practice writing this NAD structure. But what is so important about these coenzymes that we are discussing them with respect to ATP synthesis? There are a lot of coenzymes in our body. In fact, many vitamins we take are nothing but coenzymes. Either they are coenzymes directly, or they are part of coenzymes, or they are the ones which are going to help the synthesis of coenzymes. For example, NAD is a vitamin. I hope you know this niacin vitamin, right? So why is niacin required for the synthesis of NAD in the body? Now, you look at this redox reaction of NAD. Okay, this is the NAD. I have just shortened the other part, the ribose and the ATP part, and I'm only showing only the nicotinamide part here, the structure of the nicotinamide part. And the nicotinamide is interesting moiety because this can reversibly undergo oxidation and reduction. When it undergoes reduction, it takes a hydrogen here. Can you see here? If it has undergone reduction, it is simply the electron is withdrawn, bond shifts here, creating a positive center to which a hydride is added. So you get something called NADH. What is NADH? NADH is nothing but the reduced form of NAD. So NADH is the reduced form. NAD plus is the oxidized form. NADH is oxidized to NAD plus. NAD plus is reduced to NADH. Fine. Similarly, you also have FADH2, the structure of which I am not showing, don't worry. FADH2 is the reduced form. FAD is the oxidized form. We will not concentrate on this reduction direction. Rather, the oxidation direction is important to us. So, if a molecule of NADH or a mole of NADH is oxidized to get NAD plus, and I hope you know the different ways of looking at an oxidation reaction. Either the removal of electron is oxidation, addition of oxygen is oxidation, removal of hydrogen is oxidation. In this case, it's the removal of electron, is it not? So you see, if NADH is oxidized, electrons are removed. Therefore, you get NAD plus. And this cannot happen by itself. There should be an oxidizing agent which should do this. Which is that oxidizing agent? We'll see that later. As of now, we will only look at this direction of the reaction, oxidation, reduction. Now, the oxidation reaction is very much exergonic reaction. Anyways, the same reaction I have just written here in the form of a scheme, including the oxidizing agent. So oxidation cannot take place as it by itself. There should be some oxidizing agent. In organic chemistry as well as in inorganic chemistry, you come across a lot of oxidation reactions. But everywhere in any reaction, you require oxidizing agent, right? You come across a lot of reduction reactions. You require a reducing agent. In fact, the oxidizing and reducing agents are a very important group of reagents in organic synthesis. Without them, you cannot make any basic. You cannot make a lot of synthesis. You cannot make without oxidizing and reducing agent. But like we have oxidizing agents in organic laboratories, chemistry laboratories, do our bodies have oxidizing agents? Because we are also conducting, bodies are conducting, cells are conducting a lot of oxidation reactions. Definitely they must have. The principles of reactions are same, whether it is in the laboratory or in the body. You cannot do a reaction which is non-spontaneous without the application of energy. That principle is same. You cannot do a reaction without oxidizing agent if you are doing oxidizing a reaction. That is also same. So requirement of oxidizing agents are there in the body also, in the cells also. Anyways, we will see that later, which are the oxidizing agent. As of now, we will oxidize this NADH using molecular oxygen itself, which is the readily available, very inexpensive, efficient oxidizing agent, oxygen in the air, molecular oxygen. That itself is a very strong oxidizing agent. In fact, very inexpensive. But I hope that you all follow the news, right? The oxygen is very inexpensive in atmosphere. It's very freely available. The same oxygen, when it is concentrated and put into cylinders, very costly. And we have seen how the people were after these oxygen cylinders and how the shortage of oxygen cylinder led into a health crisis. Anyways, it's a different aspect of the oxygen. And as of for our discussion, oxygen is there everywhere. The cells can get oxygen just by diffusion of oxygen from the air. Anyways, so oxidizing agent. So first oxidizing agent in the cell is the oxygen itself. So if the oxygen oxidizes NADH to NAD+, how much energy is liberated? Minus 52.6 kilocalories per mole. Huge amount of energy, right? How much energy is liberated when an ATP is hydrolyzed? When an ATP is becoming ADP? 7.3 kilocalories per mole. What is the energy liberated with this oxidation reaction? 52.6 kilocalories when NADH is oxidized to NAD+. That means this is an exergonic reaction. Of course, exergonic reaction. All oxidations are exergonic reactions. You cannot have any oxidation reaction which is endergonic or endothermic. All oxidations are exergonic and exothermic. Therefore, the oxidation of NADH should also be so. Now, when it is 52.6 kilocalories per mole liberated, when NADH is oxidized, what do you think of this spontaneity and the exergonic nature of this reaction? Too much, huge. But with this huge exergonic reaction, very lot of energy is liberated, cell also sees an opportunity. What is the opportunity here? If this reaction is somehow coupled with ATP synthesis, what would happen? In fact, if for 1 mole of ATP, 7.3 kilocalories per mole is enough, 52.6 kilocalories would be yielding how many ATPs? Almost 7 ATPs, right? So with oxidation of 1 mole of NADH, a cell can get 7 ATP, 7 moles of ATPs. So a very amazing reaction this is with respect to its utility for ATP synthesis. Similarly, the other coenzyme FADH2, if it is oxidized to FAD, what is the energy liberated? 43.4 kilocalories per mole. Not as much exergonic as NADH oxidation, but still by the standards of energy requirement to synthesize ATP, it is still more. Almost 6 7s are 42, right? Almost 6 ATPs can be synthesized if you can convert all the energy liberated by FADH2 oxidation into ATPs. And if you can convert all the energy liberated from NADH oxidation to ATPs, you can synthesize 7 to 8 ATPs. So this is a very big opportunity, huge opportunity to the cells. But the actual execution of this plan is not very easy and straightforward as we are discussing. The cell doesn't directly combine NADH with oxygen. That's the first point. Fine. So oxygen doesn't directly react with NADH. In the laboratories, you can do that. You can take NADH molecule, directly react with oxygen and see that so much energy is liberated. But in cell, it doesn't happen. Rather, NADH is oxidized very slowly and in a stepwise manner. Fine. Now, why are cells far more efficient than engines which have been created by man? you have engines. What are these engines basically? Combustion engines, petrol engines, diesel engines. They are the systems which convert some form of the energy into mechanical energy, right? So they can convert heat energy, basically your petrol and diesel fuel, heat energy into mechanical energy. You can burn petrol or diesel in a cylinder and convert that into rotation of the wheels. So cells, with respect to harnessing the energy, can also be regarded as engines. They convert one form of energy into another form. Definitely, they convert one form into another. But how the energy conversion happens there and here? In fact, in the engines, it's the combustion reaction which is responsible for the energy release. Whereas in the cells, now if we are considering cells as engines, cells, it's not the combustion happen. Now that we come to a point. What's the difference between combustion uh, and oxidation? See, combustion is also oxidation, which happens at once. So, uncontrolled, rapid, sudden oxidation is combustion. So, what happens in combustion? In one step, the molecule is oxidized to its products. Whereas in cells, for example, the molecule which is getting oxidized here is NADH. Doesn't happen in one step. If it happens in one step, a large amount of energy is released, essentially a combustion, and so much energy at once cannot be converted efficiently into ATPs. Therefore, what the cells have done, they have taken a different route. Instead of single step oxidation of NADH, multi-step oxidation of NADH. So in every step, slowly energy is released. And of course, we should all recall the laws of thermodynamics. If a reaction is done in one step or in multi-steps, as long as the reactants and products are same, the total energy input and output is same, right? So whether you oxidize NADH in single step or in 10 steps, finally the energy liberated is still 52.6. If it is combustion, in one step so much energy is released. If it is multi-step, every step small amounts of energies are released. And if you add up all these liberated energy, finally it should come to minus 52.6 kilocalories per mole. That's fine. So how does the cell do this? The cell employs a very versatile, efficient, energy conserving, transforming machinery called mitochondria. Fine. So whenever you are asked the question, what is the role of mitochondria in a cell? Most of the times your answers would be mitochondria is the powerhouse of the cell. What do you mean? Why are they called powerhouses? Because they produce ATP, they produce energies. That was a very broad answer. Mitochondria is the powerhouse of a cell. But since we have studied many mechanisms chemistry wise, what is this power? So all the power will be ATP synthesis, right? At this point now, what is that powerhouse? How to define this powerhouse from biochemistry point of view? Powerhouse is nothing but mitochondria's function. They are doing oxidation of NADH. Simple, yeah, very simple. What is the role of mitochondria? They are doing oxidations of NADH and oxidations of FADH2. And the energy liberated from these oxidation reactions, the mitochondria are using those energy, coupling those energy to the synthesis of ATP. So technically, what is the way we have to define this mitochondria? Mitochondria are the site of oxidative phosphorylation. Okay, I hope you understood this term. Mitochondria are the site of oxidative phosphorylation. Now, briefly, let us examine the structure of mitochondria. In fact, since you're life science students, I think you know a lot more of mitochondria than me. But the minimum structural features of mitochondria, which are essential for our further discussions, we will see. Mitochondria is basically the oval-shaped bodies which have two membranes, the outer membrane and the inner membrane. And these outer and inner membranes are there for any cell organelle. Even the cell itself, plasma membrane, has two membranes, right? The outer membrane and the inner membrane. Now, the inner membrane is not smooth. Rather, it is folded into finger-like projections. You can see this, right? This is the outer membrane, which is in the brown color, whereas the pink color one is the inner membrane. It has numerous foldings, infoldings, projections inwards. If this is the center of mitochondria or the cavity of mitochondria, there are numerous foldings. And these foldings are called cristae. The inside of the mitochondria, inside the cristae, we have a fluid called mitochondrial matrix. And there is a space between inner membrane and the outer membrane. And the space is called inner membranous space. So on one side of the cristae, or we can call on the inner side of the cristae, mitochondrial matrix. On the outer side of the cristae, space, inner membranous space. And please remember, this is not only this is a very gross structure of mitochondria. But what is very peculiar about mitochondria, strange, is it is the only cell organelle which has its own DNA. Okay, so please remember this. Mitochondria has its own DNA. So what happens? Why is the DNA required for mitochondria? To make all the enzymes of these oxidation reactions. There are many theories how the regarding the evolution of mitochondria, how the cells got this mitochondria. Anyways, the one thing which I did not tell you is the oxidative phosphorylation happens in mitochondria. And which type of organisms is mitochondria present? Only in aerobic organisms. Anaerobes. There are species which are anaerobes also. They lack mitochondria. They do not have mitochondria. So they do not conduct oxidative phosphorylation. Anaerobic organisms. Examples, yeasts, right? All the organisms which depend on fermentation or which carry out fermentation, like yeast, lactobacillus, they do not have mitochondria. Why? Because they don't require them. The difference between these two things we are going to study later. But as of now, the mitochondria is an essential part of our aerobic organisms, cells of aerobic organisms. It's an essential part, mitochondria. And in fact, based on the energy requirement of the cells, number of mitochondria also vary. Like in your own body, all tissues do not have identical energy requirements. Some tissues have more energy requirement than the other tissues. Now, which is the organ which requires maximum energy in our body? Of course, brain. Brain requires, in fact, almost 20 to 25% of the glucose we consume exclusively goes to the energy supplies of the brain. Okay, so it's a very high energy demanding organ, brain is. Whether you're thinking or not, that's different. Whether you're using your brain or not, still the brain is the organ which requires a large amount of energy. So depending on the energy requirement of the tissues, the number of mitochondria vary. Very high energy demanding tissues, using tissues, have large number of mitochondria in their cells. Fine. As well as in those energy demanding tissues, if you take a cross section of mitochondria, the number of cristae is also more. Fine. Not just the number of mitochondria organelles, within mitochondria, the number of cristae will also be more for energy demanding cells. So that means what? The mitochondria and the cristae are directly related to energy demands. Whenever there is high energy demand, mitochondria and cristae are more. So mitochondria are the powerhouses basically now. They conduct oxidative phosphorylation. That's the answer we should give now onwards. What is the role of mitochondria? Don't tell they're just powerhouses. They are the sites of oxidative phosphorylation in cells. So the structure of mitochondria. Now we will look into the mechanism of oxidative phosphorylation in our next class. And the mechanism is called electron transport chain. Electron transport chain. So I'll just stop here, because if I start, I should continue this till the end. Only then the flows comes out and the understanding is better. So it's fine. If you have any, okay, let us spend some time for interactions also. Yeah, please go ahead, ask questions if you have one.